Thank you for joining me, Professor Soria. Tell me, why did you and your colleagues create this conference?
Well, the MAP conference, in fact, is trying to address one of the most important scientific questions that the cancer community is facing, which is how do we really integrate in daily practice the wealth of information that comes from the analysis of a tumor sample. There is many structural DNA changes that happen in a tumor, and we need to make sense of the signal we can get from that analysis and integrate that in daily cancer care, diagnosis, prognosis, and therapeutic decision.
What can we learn from the recent trials, and how will these findings be reflected in the conference program?
Well, there are many trials that have led to the registration of targeted therapies on the basis of the identification of an oncogene driver. This has been the case for HER2 amplification and breast cancer, not only with trastuzumab, but also with pertuzumab or TDM1. It's also the case in lung cancer with EGFR mutation and many EGFR inhibitors, ALK translocation and ALK inhibitors, and the list can go beyond in other solid tumors. But on top of those registration trials, there is a significant number of precision medicine trials that have happened over the last 5 years that are trying to demonstrate that holistic approaches, meaning the analysis of a tumor sample with a high throughput analysis of the structural DNA changes, can bring information that is useful to triage the patient and try to address them to different trials. So I will just take one illustration, which is the recently published trial by Fabrice André and collaborators called SAPHIR-01 Breast, that has demonstrated in the clinical setting the possibility within a network of collaborative centers in France that on-purpose tumor biopsies in breast cancer is feasible. Getting the analysis of these samples within a clinical compatible time frame is feasible, and this allows for the identification of different drivers.
So precisely, does molecular biology currently play a role in the daily treatment of cancer?
Well, as you know, under molecular biology, many things can be considered, but it is true that in daily practice, there are many tumor types for which the molecular analysis of a gene or a set of genes is necessary. So if we take the example of rare tumors like gastrointestinal stromal tumors, the GIST, analysis of the KIT mutation and the specific exons where this happens is key to decide on the therapy, imatinib, sunitinib, or other tyrosine kinase inhibitors. This will also be the case for breast cancer, lung cancer, colorectal cancer, and other important solid tumors.
Does molecular biology currently play a role in the daily treatment of cancer?
I believe so, and this is the case in many solid tumor types. Rare cancers like the gastrointestinal stromal tumors, GIST, frequent cancers such as breast cancer, colorectal cancer, and lung cancer. Let me give you an illustration in the setting of thoracic malignancies, non-small cell lung cancer, which I know better. In that setting, it is absolutely key to identify some of the frequent oncogenic drivers, because the patients who have an oncogenic driver, EGFR mutation, ALK translocation, or ROS translocation, have a better intrinsic prognosis. Median survivals are 36 months, while the standard survival for metastatic non-small cell lung cancer is 12 months. So you see there is a 1 to 3 length of survival. And the therapies are not going to be the same. In the presence of an oncogenic driver, we will be using a variety of kinase inhibitors to guide the therapy, which might not be the case in the setting of patients who do not have an oncogenic driver. This is one very practical example. So today, standard of care in lung cancer makes mandatory the analysis of some oncogenic drivers that represent 15 to 20% of the patients. Much higher percentage in Asia, for instance, where these alterations represent up to 40% of the patients. Of course, we will discuss these kind of issues during the MAP conference in Paris. We have a dedicated session on genomic drivers in different tumor types, including lung cancer with a presentation by Professor Roman Thomas. We have a dedicated presentation also in gastric cancer that is scheduled, and we have a presentation in breast cancer that is also scheduled, both by extremely talented speakers from Asia and the UK.
So what are the latest emerging technologies for personalized medicine?
Well, there are different approaches. I would say that two ways of looking at the problem. There are technologies that try to make the most of a tissue sample. And these are mostly focusing either on analyzing structural DNA changes on the tumor cells or analyzing the tumor microenvironment. That's on one side. On the other side, there are all the approaches that will not be based on the tumor tissue, but based on blood, and that's what some people refer to as liquid biopsies. So we try to characterize the cancer environment in the blood. And in the blood, not only you can analyze mutations, but technology probably is allowing us now also to look for some translocations. On top of that, we know that in the blood we have circulating tumor cells. We do have also exosomes who carry inside their vesicles DNA, RNA, and proteins. All these issues we'll also be discussing during the MAC conference. We have a dedicated liquid biopsy focus where we will be discussing the clinical applications as well as the technological features and challenges that we are facing in that setting.
How can sequencing predict efficacy of immune checkpoint inhibitors?
Well, it is interesting to realize that precision medicine and immune oncology are not two divergent paths. There is some area of synergy and potential overlap. And in fact, there are recent publications suggesting that the mutational tumor load in a given sample, meaning how many mutations you have per megabase, might predict for the efficacy of some immune checkpoint inhibitors. So the higher the mutational load, the higher the efficacy of the PD-1 or PD-L1 antibodies. And this has been demonstrated in melanoma, in lung cancer, and in colorectal cancer with mismatch repair deficiencies. So this is a very interesting topic where two different scientific themes, immune oncology and precision medicine, instead of being perceived as antinomic, really have an overlap, and there is a common theme that might help us better understand how the structural DNA changes can predict for the efficacy of immune checkpoint blockade. This is the topic of Professor Quesada's presentation during the MAP meeting in Paris.
And finally, given what we expect from emerging biotechnology, could we imagine that one day cancer diagnosis might become a routine practice with a simple blood test?
I think that's an area of a very heated debate. Our friends in pathology believe that an analysis of the tissue remains the gold standard, and for the time being, I believe that is true. Now, having longitudinal biopsies of the same disease of the same patient comes with significant challenges, correct? If you as a human being have a tumor, and I tell you I'm going to put a needle in your tumor every 6 months, you might tell me, I don't like that perspective, I don't like that idea. Liquid biopsies do provide an alternative for that, for eventually longitudinal follow-up of the molecular architecture of a tumor and the adaptation of that tumor under the therapeutic pressure. They might also represent an alternative when we look at patients at high risk, patients who might develop a cancer, heavy smokers, or patients who have a given genetic background, where becoming interventional, for instance, biopsying nodules in the lung, comes with intrinsic challenges of toxicity and cost, and maybe some blood sampling might help refine a more sophisticated algorithm, that is just based on the size of the nodule in the lung and how quickly it grows. So I really think biology, precision medicine, and hopefully the MAP meeting are going to bring a lot to the table to demonstrate that the future will in part be improved through biology-driven thinking and biology-driven medicine.
Thank you very much.